Welcome, science friends, to Frameshift Mutations. We need to know what a reading frame is before we can talk about frameshift mutations. So when we have our mRNA, it is read, so to speak, by the tRNA because the codons on the mRNA match with the anticodons on the tRNA. We read those codons as 3 base pairs at a time. When we look at those 3 base pairs at a time, that is considered the reading frame because it's always 3 and it's 3 at a time every time to code for those amino acids. Whichever codon is read first determines the frame of the codons all the way after it. So if we start off by 1, all of the other codons are going to be off by 1. So when we talk about the reading frame, that just means that codon of 3 nucleotides. Sometimes the reading frame shifts. So let's take a look at the DNA strands below. In the first strand, we have GGT as our first codon. That is the first reading frame. In the second example down below, we've shifted over 1 nucleotide. Now it reads GTC instead. That's not great, because if you look at the amino acids, they've changed. In the top example with GGT, that codes for glycine, and then the next one and the next one, they all have different amino acids. But when we go down to the second example, the frame shift, we moved over by 1 nucleotide. Now our first reading frame, instead of coding for glycine, codes for valine. And all of the other codons afterwards are affected because we shifted over by 1. And now they code for completely different amino acids than they should in that correct reading from the wild type. Because of this shift, that means everything after that shift gets changed. It's an easy way to think of everything downstream. So if you know anything about how rivers and streams work, what happens at the top affects everything at the bottom. So if I were to pour some food coloring into the very top of a river, all of the water after that is going to have some dye in it. So here's a great question. Where would this frameshift mutation have the most change? At the beginning of a gene or the end of a gene? Well, if we think about how the reading frame affects everything after it, the earlier the frame shift, the more terrible it is for the rest of the gene. So there are different types of frameshift mutations. First, there's the single base pair deletion. So we can just get rid of one of those nucleotides and that'll shift things afterwards. So here in our wild type, we can see on our third codon, we have UUU, and that codes for phenylalanine. Well, if we take away and delete that third U, well, now our reading frame has shifted. Instead of UUU, we get UUG, and everything after that is now shifted over 1. So our fourth amino acid, instead of coding for glycine, we get alanine, and everything after that is going to be changed. This is devastating. It's so bad for this protein because a change happened and that one change caused all the other things after it to change as well. We can see the same thing happening when we add a single nucleotide. So again, with our wild type at the top, we're looking at our second codon instead of an AAG coding for lysine. If we add in a U at the beginning of that codon, now it's UAA. We shifted our reading frame because we have a new nucleotide coming in. And UUA, instead of coding for lysine like we should, it's an early stop. This, again, is really bad for our protein because it's not getting all of the amino acids it needs. Everything after that is affected. The other frameshift mutation we can have is called a 3 nucleotide deletion or addition. So instead of just adding or taking away 1 nucleotide, we add or subtract 3 of them. So we're going by a codon, essentially. So in our wild type, we have methionine, lysine, phenylalanine, and glycine. That's the way it should be. If we take out that second codon, well, now we're missing an amino acid. We don't have lysine anymore. It just goes from methionine to phenylalanine. That's not good. We can see the same thing happening for an addition. Instead of removing something and things shifting to close the gap, now we're adding something and everything afterwards have to shift to make space. So now we're adding an unnecessary amino acid. Again, depending on the protein, they're very long. This could be not a big deal, or it could be really bad for our protein. Overall, frameshift mutations are worse than point mutations because we're shifting that reading frame. It could just be by one nucleotide, or it could be by an entire codon. A nice real-world example of a frameshift mutation is cystic fibrosis. By losing one amino acid, so changing just one little tiny piece of this protein, creates a really debilitating disease. So cystic fibrosis is found primarily in whites of European descent. The frequency is about 1 in every 2,500 births, and 1 in 25 whites is a carrier of this disease. The protein that's affected is a membrane protein, which we've learned about, and it controls how much of a chlorine ion can get into and out of a cell. It's a lot harder for this molecule to get into and out of the cell when somebody has cystic fibrosis. Without treatment, children usually die before the age of 5, and with treatment, people can live into their 20s, but usually they don't have a very long life. In summary, here's some good vocab words that you need to know. Wild type is a normal sequence of a gene. There's no mutations, it's just what it's supposed to be. We also have the point mutations of substitutions where we're swapping out one nucleotide. So we're not adding or subtracting, we're just swapping out and substituting. And we have silent mutations, which means no effect on the protein, missense, which changes one of our amino acids, and nonsense, which creates an early stop codon. Frameshift mutations, we have inserting and deleting, so we're adding in a nucleotide, removing a nucleotide, or we can go a codon at a time, so adding 3 nucleotides or removing 3 nucleotides. Frameshift mutations are much worse than substitution mutations because, remember, the reading frame is shifting. So everything after that one mistake or that one mutation affects all of the other amino acids in the protein. It makes a sort of domino effect or a cascade. That was Frameshift Mutations. I hope it helped.